Okay, so let's go ahead and for the rest of our time together, we have a couple of chapters to sort of discuss, and one of them is about genetic engineering, and so I'm going to combine genetic engineering with a little bit about bioethics, and so this is the first of two videos, so let's get started. So genetic engineering refers to a number of techniques for creating, amplifying, and manipulating specific DNA fragments. We're going to talk just very briefly about how to create recombinant DNA and how then to amplify DNA. And then in the second video, we'll talk about how to find a specific gene. For review, I recommend that you go ahead and go on to Google, actually, and search out McGraw-Hill for some early genetic engineering experiments. There's a number of animations that'll be fairly helpful for you. We're not going to talk about CRISPR technology. We're going to, in fact, talk about the older technology, restriction endonucleases. We will cover steps in cloning a gene and then PCR. So the end products of genetic engineering is what we call recombinant DNA. And that just simply means that we take DNA from one organism and we put it together with DNA for a different organism. Now, the two organisms could end up being the same species technically, but for the most part, what you should think about is that we're taking DNA from an individual, a mouse, a human, a goat, a cow, a chicken, what have you, and then replacing it into bacteria or yeast in order to grow copies of that. Now, gene therapy, which we'll talk about just briefly in the second video, is obviously the idea of using technology to replace or enhance or do something to the genome directly, all right? So, but anyway, recombinant DNA is simply two different DNA molecules from two different sources, right? So some famous examples, you should probably be aware of the green fluorescent protein gene that can be incorporated into pretty much any host organism. And so if you go to an aquarium shop now, you can buy fish that have been genetically modified so that when you give them a UV light, they'll glow in the dark for you. All right. So at any rate, the term here is transfected when you use a gene, put it into a host. Okay. So there's lots and lots of different technologies now. Beginning in the 1970s is where the historical reference point. So things that we identify from nature, we then modify to enhance their properties. But so the restriction enzymes, ligase, polymerases, proteases, and now, of course, the CRISPR technology, all trace from things that are found in nature, right? So things from nature allow us to clone, to do PCR, the various flavors of PCR, sequencing, fish technology, microarrays, you name it, right? So how do we take all of those different techniques and sum them up? I've mentioned this a couple of times in class, and so really, if you think about it, there's just two fundamental principles of molecular biology. A whole bunch of varieties and flavors that derive from these two principles, but the first one is DNA, RNA, nucleotide sequences, of course, obey base pair rules, right? And so they form hydrogen bonds, A with T, G with C, and so on. And so you can exploit that by designing sequences knowing that where A's are present, T's will form hydrogen bonds, right? So this is a very powerful principle, the idea you can design sequences and you know what the match will be. So the other principle is the idea, the concept that proteins recognize specific DNA sequences. And in recognizing those sequences, right, those DNA elements, okay, they affect their function. So transcription factors are famous examples of proteins that interact at specific, bind to and interact with specific DNA sequences. Okay, all right. Okay, so just jumping into the idea of how to amplify, make copies of DNA, there's two methods, and one of them is cloning. So cloning is a procedure of isolating a known DNA sequence and then making multiple copies of it in vivo using a live organism. So the live organism that you're using to make copies of your DNA is called the vector. Okay. Minimally, what the sequence has to be linked to a regulatory sequence of a gene in the vector in order for it to be, you can take advantage of the copies, right? So, all right, the other way to make copies of DNA, of course, is polymerase chain reaction. So PCR in principle can be performed on any DNA, and with PCR it's possible to go from a few copies to millions to billions of copies. Just like cloning, you go from a few copies to potentially millions to billions of copies. The difference, of course, is that the copies are now, in cloning, the copies are now in a vector or in a living organism that you're going to have to then extract and purify your sequences, whereas PCR, of course, the end result is pure sequence, copies that you want. So just a very brief history. It's like most things, somebody gets credit for it, and of course immediately people point out, hey, what about me? I was involved. So at any rate, sort of the standard story is that Kary Mullis gets credit for inventing PCR, and his key innovation was the use of Taq polymerase from an organism that's found on thermal vents. So it's heat tolerant, and so by incorporating a heat tolerant DNA polymerase, it revolutionized the idea of how to make copies of DNA. All right. So basic highlights of DNR involves the three steps, right? The initial steps are to denature double-stranded DNA at a temperature close to boiling, and then you cool it, the annealing step, by lowering the temperature anywhere from about 35 to 65 degrees Celsius, and we'll kind of return back to that in a moment here. And then the third phase is, of course, to make copies. And so we warm the thing back up. 72 degrees is the temperature the Taq polymerase likes, Taq and its variants. And then this is where the nuts and bolts of PCR, making copies of your DNA, are affected. So you have your forward and reverse primer sequences, you have your deoxynucleotides, the triphosphate versions, of course. You have your polymerase. There's now lots of different polymerases. You have magnesium, which is a cofactor and of course you need a thermal cycler under computer control to regulate the temperature as well as the cycling phase, right? So I mentioned we come back to the annealing temperature, and this is one of the ways that you can manipulate specificity. So at colder temperatures, pretty much any sequence that's close to your match will anneal, will bind up, form complementary base pairs, but it won't have to have 100% identity. So you can have mismatches, and that's all right. Thermodynamically, it's single-stranded DNA much prefers to be double-stranded DNA, and so even if the match isn't perfect, you're going to get double-stranded DNA at 35 degrees. At the higher temperatures, this favors specificity. So in order for the two strands to come together and not separate at that very warm temperature, you're going to have pretty much 100% matching between your A's, T's, G's, and C's in order that the maximum number of hydrogen bonds are present. Okay, so sequence specificity certainly by design is the intent, but you can also play around with the thermal cycling conditions, particularly the annealing temperature, to favor the products that you want. Okay, so PCR, you're probably much more familiar with PCR than you are with cloning. And some of you in lab have done some cloning. We're going to do a little bit of cloning work next semester. So at any rate, I thought I'd throw this in here. GMO, what's GMO? GMO involves taking some donor DNA, putting it together with your vector DNA, and then whammo, you now have a genetically modified organism, right? Okay. Sample of DNA with the gene of interest. It could be from genomic DNA. One thing that you do is to make complementary DNA, which is you find messenger RNAs, you reverse transcribe it to DNA, make it double-stranded DNA. It's now complementary to the messenger RNA that's present, so you can have a cDNA library, which represents, of course, all of the expressed protein coding genes. And of course, you can just make up your own DNA, right? Any of those could be the donor DNA of interest. And then your choice for vectors is also quite various now. And so basically the idea is that many organisms, you can insert a non-essential chromosome, and you will then be able to make copies of the DNA that you want if it's inserted into a non-essential chromosome, because when the cells go from one cell to two cells, etc., they make a copy of all the DNA that's present. Right? Okay, so the choice of the vector DNA is basically, well, how big is the host, the donor DNA? If it's really small, then plasmids, which are small circular independent chromosomes ranging up to about 30,000 base pair, you could maybe insert your DNA into that. You might use bacteriophage, which is, of course, a virus of bacteria. And these have pretty small chromosome sizes as well. And then there's cosmids, which are a combination of phage and plasmid DNA, and we start getting larger and larger now, the vector DNA. Artificial chromosomes that have been manufactured, bacterial artificial chromosomes, this was for plasmids, okay? And then finally, we now have the technology to insert artificial chromosomes, pretty large size, into yeast, okay? A eukaryote, right? Okay, so an example is you've selected some DNA from a genomic region, and you can target it, well, by CRISPR technology, or the older ways would be to use restriction endonucleases. So restriction enzymes are derived from bacteria species. Each species has its own, and there's hundreds of these enzymes now known, and they're specific. They recognize a specific DNA element, and they bind to that specific element, and then they cut at that specific element. And they cut in a variety of ways. But again, the idea, just like CRISPR, is a more precise variant of this, but it's the idea is that you're going to know ahead of time where your enzymes are going to cut. Okay? So restriction sites can range from 4 bases, the message itself, 4 bases long, 6 bases, 8 bases, or even more. And the idea then is that if you're using a 4 cutter, it'll generate on average about a cut every 256 bases. A 6 base cutter, about every 4,000 bases, an 8 base, okay, about every 65,000. So anyway, it gives you a sort of size description. Okay, what restriction enzymes do is that they give you a cut that allows you to have an overhang for one, so we call those sticky ends. And so this is an example of an EcoR1, the message that it recognizes, and then it cuts between the G and the A, and between the G and the A in the 5' direction, right? This particular sequence is called a palindrome, right? Because it's a reverse of itself. All right. So there's my restriction enzyme. Just had some cutting going on. And there's our sequences. And notice we have now the overhanging fragments. Okay? So what you would do is with your donor DNA, you'd use the same restriction enzyme as with the vector DNA, and then when you add the donor DNA plus the vector DNA, the idea is that these different DNA sources have complementary overhangs that'll allow them to stick together. Pretty cool. All right. So that's that idea. If you digest both, then you're going to have some complementary ends. You add those together with DNA ligase, and the result will be you're going to have, of course, a bunch of DNA that is a mixture between the two sources. All right, and so then you'll also have some DNA that, of course, is single source, where the original DNA just snaps back together and doesn't incorporate the different one. So you're going to need a technique now to separate that. And so where the business end of the cloning is often involved in doing some selection. Okay, so because you want to select certain vectors that have your recombinant DNA. Okay. So your vector, your plasmid, for example, will have a site where you're going to insert the DNA, but then it'll also have a couple of genes that will allow you to then grow the bacteria in different media conditions. So, for example, you might have a bacteria or antibiotic resistant gene, and then another gene like beta galactosidase, which would mean that it can break up lactose, right? Okay. All right. So you're basically going to transform the bacteria. That means you mix your recombinant plasmids with your host cells, and you're going to select your transgenic cells by putting them on different media. Okay. So the way that one type of plasmid works is that you'll have the site where the recombinant DNA is going to come in disrupts the antibiotic gene. Okay, and so you're going to grow your DNA inside those colonies on an ampicillin-resistant plate, basically a selector. Okay, so for example, the blue-white technique. All right. All right, and so then you can transfer them to a membrane, blah, 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 blah. But the point is at the end of this, you're going to be able to tell, which bacteria have your plasmid, because those that have your recombinant DNA, you grow them on the ampicillin plate, they don't grow, and so that means that they have the antibiotic gene disrupted. So you just go back to where you were making copies and you can identify the colonies. Okay, so that's the end for this video, and stay tuned for the next video.